Hello, everyone. My name is Michael Key. I'm the CEO of Impello Biosciences. We're a biostimulant company located in northern Colorado. And today we'll be talking about how we can manipulate environmental inputs in cannabis cultivation to optimize genetic expression of that cannabis variety. So that being said, it's a pretty broad topic. I'm not sure how familiar everyone is with biostimulants, but they're kind of a subclass of agricultural or horticultural inputs. And when we talk about or environmental inputs as a whole, of course, there's a lot of different factors that we can manipulate. If you're in a controlled environment, you can obviously control a lot more of those variables than you can if you're, let's say, growing outdoors in a field. But regardless of the environment and the situation that you're in, focusing on your environmental inputs is a very important way that you can control and kind of dictate your crop's performance. So we'll get into all of that and how they relate to biostimulants. But the four overarching ideas that I think we'll cover over the next 30 minutes are as follows here. So I like to remind people, and I always like to think about plants' environment and its environmental limitations as opportunities. And that's a unique perspective to have that we're afforded only by working with biostimulants. So to give an example, in a cannabis environment, let's say we're dealing with a soil type that may be historically subpar or not optimum for cannabis cultivation. But maybe over seasons of growing, we've seen some really unique plant phenotypes or really unique plant flower qualities coming out of that. Instead of looking at that soil now simply as an environmental limitation, we can actually use biostimulant technology and use our knowledge of the environment to dive into that soil, use sequencing techniques, look at plants' genetic expression or its transcriptomic expression, its metabolomic expression, to actually understand what characteristics or what aspects of that soil are providing some benefits and then what's not providing benefits. So instead of just looking at something like a subpar soil as an environmental limitation, we can actually start to exploit the good aspects of it and then figure out how to eliminate the bad aspects. That kind of plays directly into the idea that biostimulants are a new variable in crop cultivation. So historically, we've had fertilizers. If you're growing inside, or some people outside use carbon dioxide injection as a way to improve plant productivity. Of course, we can control irrigation, but biostimulants I look at as a new variable in a plant growth equation, a biological equation. So if you're adding a biostimulant that's capable of promoting growth through different mechanisms or in a different way than a traditional fertilizer or a pesticide might be able to, we're actually increasing the theoretical limit of plant productivity that would otherwise be limited simply or constrained by the types of inputs that we're using. So using something like a biostimulant will allow us to, again, increase that theoretical plant potential maximum and actually achieve, whether it's greater yields or higher plant quality or both, achieve something that would otherwise be unachievable by just using traditional ag inputs. Now, again, that funnels down into the third overarching idea here, and that's that biostimulants not only are a new variable and act in a new way, but they do that on a scale that's pretty atypical to previous generations or iterations of agricultural inputs. And what I mean by that is it has a whole omic input or influence. I didn't create this term, but it's something that we experience a lot in my company. So while we started, we focused primarily on the metabolomic influence of different crop inputs. Being in the cannabis industry, that's one of the easiest things to focus on. Of course, most hemp or cannabis growers are testing for secondary metabolites like cannabinoids and terpenes, and looking at how the inputs that we use are influencing the production of those metabolites is important. But we realized pretty early on that these biostimulants are so powerful and have such an overarching influence on crop performance that not only are we influencing metabolomics, but logically we're actually can work backwards and say, well, we're influencing the proteome. And before that, we're influencing the transcriptome. And even on a genomic level, these biostimulant inputs are having an effect. So again, that plays into the whole idea that a crop's genetic potential is kind of the basis. And then we can manipulate the environment to have an influence from a genomic level to a transcriptomic level to a proteomic level, and then ultimately also at the metabolomic level, which is kind of the end result that we see there. So those three ideas, I think, encompass a lot of the technological side of biostimulants and how we can utilize them to optimize genetic expression in plants. But they all also play into a fundamentally important idea and concept for us to all work with, both in agriculture and in other aspects of global industry, and that's sustainability. In today's day and age, it's not a surprise that I think all new technologies should have some focus on sustainability, and biostimulants play very nicely into that. We're working with a technology here that to some extent is an alternative to genetic engineering. Of course, there's been some backlash in years, despite all the tremendous benefits that genetic engineering has provided. The perception is not quite what it used to be. And industry and consumers are actually looking for alternatives to genetic engineering solutions. And biostimulants are a very viable option for that. Not only are they highly technical, not only can they achieve a lot of things that genetic engineering can, and also some things genetic engineering cannot, but they can do it in a very sustainable, natural way by basically harnessing natural potential, nature's potential, things that are already occurring in life and in biology. But we're developing the ideas, we're understanding the mechanisms in a way that allows us to manipulate that better and make greater progress by using these sustainable technologies and have a big influence on crop productivity. So that being said, if we think about agriculture as a spectrum of ideals, and I always like to say organic farming, organic agriculture is certainly at one end of the spectrum. Then conventional or intensive agriculture would be at the other end of the spectrum. Conventional, you could say, is the black end of the spectrum, and organic is the white end of the spectrum. That being said, those two groups don't necessarily always agree on their ideas. I think there are a lot of people in the organic or at the organic end of the spectrum that are staunchly opposed to any conventional agricultural techniques. There are a lot of conventional agriculturalists or farmers who are not at all interested in incorporating organic techniques simply because of the negative stigma and the kind of thought that, well, organic is nice, but it's really impractical. That's an old idea. That's not something that I think we should carry on into the future. And that's why, as simple as this looks, I think, and as simple as this idea is, I think it's very important that if we go along with that idea that organic and conventional are at totally polar opposites, all of the gray space in between that should be looked at as a sustainability aspect. So we can take some of the best aspects of organic farming, some of the best aspects of conventional farming, and combine them in new materials and methods that ultimately are more sustainable, both on the manufacturing side and also on the production side. That's where biostimulants fall. I think it's kind of both a great characteristic and a great opportunity of the biostimulant industry to be able to play in that middle ground, where we're not making organic farming and biostimulant farming synonymous, but rather a biostimulant input can be both organic and conventional, or it could be conventional or organic, I should say. And being a biostimulant farmer doesn't necessarily mean that you're subjecting yourself to only organic practices or only conventional practices. Rather, that you're focusing on sustainability, you're focusing on utilizing the best technologies from both sides of the spectrum, all aspects of or all parts of the spectrum. And by doing so, you're actually achieving something much greater than what you would if you were just at one end or the other. So that's one ground rule. I think another ground rule to keep in mind, just as I'm going through this, is the idea, and this is an equation people have probably heard before, but that crop performance is ultimately dictated, or ultimately it comes down to a simple equation of genetics times environment. I've also heard this described as genotype by environment. The idea being that you can have tremendous genetic potential in a crop. You can have a beautiful environment, whether that's a controlled environment or a greenhouse or just optimum conditions out in a field. But you really need to have a combination of both the genetics and the crop environment for that crop to perform maximally or optimally. That's obviously the underlying idea of this presentation, but we're not going to focus so much on the genetic aspect. Of course, as a biological input company or a biostimulant company, we're particularly concerned with the environment side, but that doesn't mean that we actually don't focus quite a bit of our resources on the genetic side. We work really closely with some hemp genetics manufacturers, both industrial varieties and high CBD horticultural style crops, because we think it would be a fundamental flaw to develop biological products and biostimulant products, any sort of environmental input, without closely considering the genetics of the plant that we're developing those inputs for, because they go so hand in hand. You can't really remove one half of the equation and expect to have a full comprehensive solution. It doesn't work that way. We have to consider genetics, even though we're an input company. And likewise, genetics company should consider what's possible, what an input company is capable of producing, what farmers can actually use. And ultimately, that will help make sure that both the direction of genetic progression and the direction of biostimulant technologies are working in a way that is productive for everyone involved. Now, the last little ground rule that I would like to lay is kind of some background on biostimulants. So I think for people who have been in agriculture for a long time or who have been in academia and studied biostimulants or biological technologies for years will understand that we're not in the infancy necessarily of biostimulant technology. It's more appropriate to say we're maybe toddlers. Biostimulants have been researched for 2 decades or more. Of course, they've gained a lot of popularity in the 2000s and the 2010s, where we started to look for alternatives to genetic engineering, like I said, and we're starting to really focus on how we can increase crop productivity in ways that we haven't been able to before. We've got these tremendous demands of feeding 10 billion people by 2050. We have tremendous amounts of food waste. We grow a lot of food, we grow a lot of calories, but that's also led to problems of its own. So biostimulants fit into this nice, it's fortuitous timing to an extent, that there are cultural movements that are encouraging the research and the development and the production and the use of biostimulants. But it's also important to consider that there's huge, global demands that consumers aren't necessarily thinking of, but that us as academics, researchers, industry folks need to consider and need to start preparing for the future, more or less. So that being said, biostimulants of today are not what they were 20 years ago. So I'll say something like yield is easy, and I don't necessarily mean that in doubling or tripling or quintupling a yield is easy. It's not. What I do mean by that is 20 years ago when we had our first bacterial isolate products and our first biostimulants, there were a lot of claims made like we would increase yields, 10 or 15 or 25%. And you could replicate that in the lab, but you could never replicate that elsewhere. We've come a long way over the last 20 years. And actually, most biostimulant products, I'll take microbial, plant growth promoting bacteria, for example. Their development and their technology is so far superior nowadays to what it was 20 years ago that it's actually not surprising at all when a farmer who's not using a biostimulant or a microbial inoculant incorporates that product for the first time for them to see a 5, 10, 15% increase in yield, particularly if it's into a system that's never had any sort of input like that before. There's a replenishing aspect, especially out in the field environment, where adding even something as simple as a growth promoting bacteria can have these tremendous effects. Also can have tremendous effects on fertilizer reduction. So we've seen farmers reduce fertilizer rates by 10, 20, 25, 30% in some cases, and without any yield loss. That's not necessarily the direction that biostimulant technology is now focused on. I think that's hugely important, but biomass yield in and of itself is not necessarily the only direction, the only thing that biostimulant manufacturers and farmers need to be considering when they're using these technologies. Instead, it's coming back to that genetic potential. And cannabis is a great example of this because phenolics really are the future. So when we're developing our biostimulants at Impello, what we're focused on doing is finding biostimulant technology, microbial inoculants that increase yield, which should be pretty easy, but also increase the yield of not just biomass, but let's say phenolic compounds. When we're growing cannabis, that's ultimately what we're growing cannabis for. And I think other crops will start to revert back to that. The idea that an apple 100 years ago had about 5 times the nutritional value of an apple today tells me that we shouldn't necessarily just be focused on growing more. We should be focused on growing better. Instead of trying to grow 5 times more apples into the future to feed the whole world, we could focus on making each apple more nutritious. It's a lofty goal, but I don't think it's impossible, particularly considering the way that biostimulants often work, and that's multiple modes of action. So being biological products, being living organisms, being signaling molecules that often play multiple roles within a plant means that we have multiple different mechanisms that we can exploit out of each technology. So for example, Tribus Original is a bacterial consortia that my company makes. It's a tremendous growth promoting bacteria because each bacteria species that we incorporate is fabulous at mobilizing nutrients, solubilizing potassium or mobilizing phosphorus, fixing nitrogen even to an extent. But the benefits of a consortia or of adding a consortia don't stop at nutrient mobilization. We have all these other direct and indirect mechanisms of growth promotion that mean that something like Tribus is not just or not necessarily a jack of all trades, but it certainly is a master of more than one. So, the end idea here is that yield should be looked at as a low-hanging fruit, but there are many other higher fruits that we should focus on as we're doing biostimulant development and incorporating these products and remembering what they're really good for. So that being said, now we'll get into some more concrete examples. And I look at biostimulants, I think you can divide it into basically 3 tiers of products, at least in today's day and age. So fertilizers, I look at as a tier 1 biostimulant. Microbial products, I look at as a tier 2 biostimulant. And biochemicals are what I would consider the 3rd tier of biostimulants. So let's get into that. Biostimulating fertilizers. I think the general idea here is that they're relatively simple to use, and the goal should be to kill 2 birds with 1 stone, to put it bluntly. So we can utilize a combination of organic fertilizer, let's say, that may be biostimulating. A good example of this is a hydrosate product. So there are vegetable-based hydrosates. There are animal-based hydrosate products like fish protein hydrosates. My company produces one of those. And these products are biostimulating because they provide nutrients, which basically makes them a fertilizer. But in addition to that, they provide biostimulating compounds like organic and amino acids. Now, to give one very simple example that's relevant to hemp farmers around here, there are some hemp farms who have planted the same genetic variety for 3 or 4 years now and have had different plant responses, different plant yields every year because there are some factors of the environment that they can't control, like hail or cold temperatures. Maybe it's too wet to harvest in the spring or in the fall, or too wet to plant in the spring. One of the things that we've noticed helps buffer these variances that are out of our control are applying something like an amino acid fertilizer, or a fertilizer that contains amino acids. And when we do that, what we can do is supply the plant with some readily available amino acid compounds. So we save a plant an energetic step that it might not be able to do given its environment. Again, to use the cold, let's say a cold, rainy spring, it's cloudy outside, plant's not photosynthesizing efficiently, it's too cold to effectively take up nitrogen. We don't want that crop to stall out in the ground. So what we can do is apply a foliar application of a fertilizer, let's say a fish hydrosate, that has these amino acid compounds, provides an immediate boost of building blocks that the plant can use and doesn't have to build itself, saves it that energetic step, keeps crop productivity high, and keeps the plant ready to accelerate growth when conditions improve. On top of that, when we add biostimulating fertilizers, we're often adding carbon compounds to soil systems or to a root system, which of course encourages microbial activity. And that's another way that we can stimulate plant growth, as well as improving soil quality, which of course has a sustainability aspect to it as well. That said, moving on to the second tier of microbial inoculants, I do like to say that fertilizers are tier one because they're usually cheap. They're usually pretty easy to apply. They can be applied in conjunction with a lot of different inputs. And microbial inoculants are not so much different, but they often do bring a higher price tag. They can be a little bit more finicky to deal with. You need to be cognizant of what microbes you're adding, why and when and how. So that's why I bump them up onto a second tier. That being said, you can use any of these tiers by themselves, or you can use any of the tiers in combination with each other. And frankly, of course, if you're trying to be a truly biostimulant farmer, you'll want to use as many biostimulant products as you can. That being said, microbial inoculants have come a long way over the last 20 years. Instead of using our single isolated strains, nowadays we use bacterial consortia. My company focuses primarily on bacteria, but I'll touch briefly on fungi and then biopesticides as well. But to start with, we'll dive into bacterial products a little bit more. And I think bacterial products have a tremendous amount of potential, again, to be biostimulating, to help improve crop quality and particularly flower and fruit quality, nutritional content, things that we haven't necessarily focused on in the past, but in relationship to something like cannabis and in relationship to the Feed the World movement and sustainability movement, bacterial inoculants are starting to show how important they really are. So things to keep in mind when you're looking at inoculants is something like an isolate versus a consortia. So the old school of thought was we could use a single isolated bacteria to accomplish a single thing, like let's say as a fungicide. That works on occasion, but it doesn't work as well as it should, particularly in a real environment, let's say in a field environment. What we have figured out over the last 20 years, and something that my company is particularly fond of, is using bacterial consortia. So we use a team of bacteria that help each other thrive and actually accomplish more by working together with each other and together with the plant. And that plays directly into plant growth promoting rhizobacteria, or PGPRs. As important as the idea of a consortia is the constituents of that consortia. So we don't want to necessarily just throw a bunch of random microbes at a plant and hope that some of them are growth promoting. Instead, what we do is use new sequencing techniques like next generation sequencing, where we might look at a specific unit of rRNA, like the 16S subunit, and do a lot of DNA extraction out of rhizosphere soils, for example, which requires tremendous amounts of data analysis as well. But in the end, we come up with a combination of bacteria that not only work well together, not only are naturally abundant, but are also things that the plant is selecting for because of their growth promoting abilities. So that differentiates a growth promoting rhizobacteria or growth promoting bacteria from something like an effective microorganism that might be very good at cycling nutrients, but isn't necessarily something the plant desires to have a very close relationship with. Fungi can kind of fall into that category too. Of course, there are a lot of bacteria in that category, but fungal inoculants they have their place. They're tremendous for the most part at promoting growth during vegetative phase of plant life. But plant growth promoting bacteria seem to play a bigger role during fruit and flower ripening and the reproductive stage of life. Fungi also need to be considered in terms of their ability or their potential to be parasitic to a plant. When we use free-living microorganisms or rhizobacteria, they're happy to live on their own. They don't necessarily need a plant around. The same can't be said for a microbe like mycorrhiza. Mycorrhiza requires that plant relationship, of course, and growers often use them, or I see growers often use them in artificial environments. So an environment where they may be running a tremendous amount of supplemental phosphorus. In that case, mycorrhiza might not actually be providing a biostimulating benefit, and it might not actually be helping a plant reach its genetic potential. Instead, it's requiring a certain amount of energy from the plant to survive. So it's taking carbon resources from that plant, sugar, basically. But in turn, it's not providing the benefit that it should be, which would be phosphorus mobilization in most instances. just something to keep in mind. I'm not saying that mycorrhiza shouldn't be used, but it needs to be used with caution. Same thing goes for trichoderma. It's a tremendous nutrient cycler. It's a fairly effective biopesticide, but it needs to be used with caution because in high salinity environments, it has been shown to reduce yields. The same sort of caveats can be applied to biopesticides, where there are a lot of biopesticides that work tremendously indoors or in very specific environments. But again, this goes back to the idea that we're looking for products, for biostimulants particularly, that can enhance a plant's genetic potential and don't necessarily just serve one purpose. We want these products that we're using to serve a multitude of purposes. So we're seeing biopesticides move in that direction, where we're actually working with consortia of growth-promoting bacteria that simultaneously act as highly functional biopesticides. And that can actually be one way they promote growth. There's a lot that you can go into on that side of things. And there are a few leaders in the biopesticide development side of the industry, like Marone Bio Innovations and Bio Consortia, produce some very effective products. And they're trying to accomplish more than one thing with those. So that being said, I think it's important to always put a face to the name. The picture on the left here is an example of a small cannabis cutting, only about a week or less than 2 weeks old, with some pretty impressive root growth. Of course, it's not anything out of the ordinary necessarily, but I think the root branching is actually quite nice. And then the picture on the right is not one of our pictures, but I think it gives a good kind of graphical representation of what bacillus colonization of a root surface looks like. And that's one of the most important things to consider with these biopesticides or with growth promoting bacteria is rhizosphere competence and their ability to colonize plant tissues before anything else does. Because ultimately, when we're having all these metabolomic responses that are not necessarily just due to nutrient acquisition, it may be in response to what the microbe is doing on different plant tissues. And now that gets into, I think, the most fun part of microbial product development, biostimulant product development. Nutrient mobilization is great, but more importantly, as it relates to phenolics, we can look at how microbes, when they're associated with different soil types, let's say, or in the case of grapes and wine with different terroirs, we can say, and some groups of researchers who spent a lot of time on this, like Bokelich, who put together this very nice image of identifying different bacterial communities that are associated with different types of Californian grapes during the first stage of the winemaking process, which is like when you're mashing grapes together and making a grape must. So the bacterial communities on that grape must is distinct between the Napa and the Sonoma and the Central Coast Valley. That's fascinating. Their focus was primarily on fermentation, but going a little further, Gilbert and others put together a more comprehensive look at bacterial communities that are associated with different tissues of the grapevine. They postulated that not only do different bacterial communities influence fermentation, but actually different microbes and the microbiome of different grape cultivars is influencing that grape's flavor and quality characteristics throughout its entire life cycle. So whether it's bulk soil or rhizosphere soil or bacteria that are found on flowers or leaves or on the bark, all these microbes play a role in a plant's response. They interact with the plant and they elicit certain responses from the plant that we can't get with just fertilizers or pesticides alone, and that ultimately we'd love to be able to exploit and be able to utilize some of our knowledge that we find and that we have found and that we'll continue to find on how these different bacterial communities are associated with different terroirs, let's say, or flavors or quality characteristics, and ultimately turn those into products that we can use and replicate results elsewhere. Now that also takes us into the third tier, and that's biochemicals. So biochemicals are extremely complex. They're at a very early stage, but this is a part of the biostimulant industry and product development market that Impello is putting tremendous amounts of our research and development resources into. So real briefly, because a lot of this information is proprietary and we're working hard to develop it, but we can't share too much right now. Biochemicals, the gist is that we can use some of these as immune potentiators we can use some of these signaling hormones or other defense-related compounds that plants naturally produce to more or less trick a plant into doing what we want it to. Cannabis, again, is a great example of this because it's a plant that's devoting almost 30% at times of its energetic resources towards the production of secondary metabolites, cannabinoids, terpenes, flavonoids, silvanoids, etc. When we're farming a crop entirely for those secondary metabolites, not for biomass, but rather just for secondary metabolite content, being able to increase the production of those at all is obviously highly valuable. And as you'll recall from the start of this presentation, that boils down to the crop's genetic potential and the environment. So with these biostimulant technologies and these biochemicals, what we've been working on, and this is some of our field data from a trial this year, is utilizing different combinations of, let's say, microbial-derived biochemicals or plant-derived biochemicals or both in exogenous applications to elicit very specific responses from the plant. This graph here shows that we have we did a field trial in collaboration with Sunrise Genetics, our hemp genomics company that we work very closely with. And more or less what we found is that metabolomic response to this first iteration of biochemicals we're creating is highly dependent on the plant variety. At the bottom here on the x-axis, you see all these FL varieties. Those are different first light varieties from Sunrise. And you can see more or less, as denoted by the red, the treatment plants responded pretty similarly among all the first light varieties to the biochemical treatment that we applied, whereas some non-first light varieties that are genetically different responded quite a bit differently. And what I can't show but will tell you is that if you break down, this is a graph of total terpenes, but if you break this down into individual terpene constituents, so major and minor terpenes, we actually had massively different responses, even between varieties and especially between types of metabolites that we were looking at. So monoterpenes versus sesquiterpenes, etc., all responded very differently. We can go even further, and I can give another example to show how complicated this biochemical problem is, but how much potential there is in it, and show that this is some work we did with Colorado State University's proteomic metabolomics facility. They have incredibly precise capabilities there for looking at metabolomic and proteomic data. And so we ran a non-targeted, semi-quantitative hemp metabolite assay with them. We did not volatiles and non-volatiles. Of course, used different machinery for both. This is one of the principal component analyses graphs that we got back from the UPLC data. It's the same biostimulant applied at different temporal points throughout the plant's flowering period. So at the same concentration, but just applied at different frequencies and different time points, we elicited completely different metabolomic responses. And this is a combination of over 2,500 different metabolites, many of which we're still annotating because the system does not know the taxonomy of the cannabis system that we're using. So the point is just to show that even with a little bit of manipulation of a single biochemical or of a single combination of multiple biochemicals, we can elicit these massively different responses from the plant. And obviously that's something we need to dive deep into, particularly when considering the pharmaceutical aspect of cannabis, but also when we're looking at improving quality of other crops, grapes, hops, coffee, anything with flavor, so lots of herbs, etc. So that being said, I hope the last 30 minutes gives you a better understanding of how biostimulants can be used to influence a crop's genetic potential and maximize that potential, and how intricately related a crop's genetics are to its environment, and really how we shouldn't look at genetics and environment as two separate pieces, but rather as how we can utilize both genetics and environment, develop products to make the whole greater than the sum of the parts. And I think within all of that, hopefully you have a good understanding, or at least a good basis for understanding that biostimulants are certainly a sustainable way to pursue new technologies and agricultural inputs. Appreciate all your time, and look forward to speaking with any of you individually if you have questions please reach out to me directly via email, and I'll get back to you as soon as I can. Thanks very much.